So this was our last class where I had told you the various examples of the where RDT has been used in the field of agriculture. So I had told you about the development of BT containing crops which are insect resistant and the technique used. Then I told you that apart from cloning in the nuclear genome, there is also a possibility that you can clone your transgene in the chloroplast, with the major advantages being that you can have more copies of the transgene, and also that if you clone your gene in chloroplast, then there are chances that the gene will not spread into the environment because the pollen doesn't have chloroplast, and so it cannot be passed on. And third is that the gene can be cloned in the form of a operon if you want to clone it in chloroplast. So if you want to clone two, three different genes under control of a single promoter, you just have to do one cloning and not multiple genes with their separate promoter that you had to do if you wanted to clone it in nuclear genome. Then I had also told you that which are the various strategies by which you can counteract, you can at least avoid the development of resistance in the insects, in which I told you the one option is you can grow two toxins together. Means you can make transgenics which are overexpressing two or three different types of toxins, so that if the insect becomes resistant to one toxin, at least it is sensitive to the other one. Or you have a temporal control over the production of these toxins, or it can be in a particular organ dependent manner. Means you can design which type of promoters you have which get activated only in a particular tissue and at a particular time, so that the insects are not always exposed to these toxins, and then develop strategies to resist the death. And third is that you can also go for mixed planting. Then I told you the two strategies by which herbicide resistant crops were developed. This I told you. Then we had come to this gene inhibition approach, where I also stressed to the fact that one, that obviously that if you want to make transgenic crops, many times you might have to decrease the level of a particular gene. So that can be done by gene inhibition approaches, in which you design an antisense RNA against a particular gene. So, or you can design a siRNA or shRNA, or now we know that you can also do CRISPR-based genome editing to make transgenic plants which have altered expression of a particular gene. While telling you the example of this flavor saver tomato, or let's say making the tomatoes which avoid the spoilage by being more sturdy and not going for a quick ripening of these tomatoes, we also understood that to achieve one particular phenotype, in this case that we don't want the fruit to get ripened very quickly so that it gets spoiled before reaching the market. uh, so the final thing is same, but the strategies can be so different. What you have to know is that which are the various pathways which are involved in leading to fruit ripening. So in that context, we understood that one way is that you target a gene, that is polygalacturonase, which affects the pectin. So means one transgenic was made in which this particular gene it was inhibited, and then we got a flavor saver tomato, and which had means much slower ripening as compared to the control one. Or you look for inhibition of ethylene, that is the hormone which is involved in the fruit ripening. So in this, again, various strategies were used. In one case, they had used, inhibition of ACC synthase, which is involved in this process. One type of transgenic tomato that is in the market is based on that. In another one, what they had done was that they had introduced a gene called ACC deaminase, which will break down ACC, and so the this ethylene synthesis will be affected. And in third case, what they had done, that they had introduced this hydrolase, which reduced the level of SAM, which was a precursor to ACC. So means in some cases you can use a single gene inhibition. In some cases you can use two gene inhibition. In some cases you can overexpress one gene along with inhibition of another gene to achieve this final output. So so in that context, I told you that please go through. There are two links given. One is like development of blue rose, and another is development of some transgenic maize, where you can understand that to achieve a particular phenotype, what all strategies they had used. See, another example is that to delay the ripening of tomatoes, another strategy that was used was totally two different genes, both of which were involved in the ripening process. And very successfully, these transgenic tomatoes have been made using this strategy as well. So your mind has to be very, very active, and you have to figure out what genes to alter to achieve a particular phenotype. Then certain examples were given. I'll not go through these again. So today's class majorly is to discuss the safety concerns associated with the transgenic plants. And in this slide, I just want to tell you that very small number of crops have yet been altered to make means, or let's say genetically modified to achieve a particular phenotype. And also the crops which have been made means, let's say many transgenic crops have been made in the labs, but many of them have not yet come to the market. They may have come in some country, but in other countries they are banned and so on. So obviously it means that there is a safety concern with respect to the transgenic plants. Before coming to that, I just tell you that in India, it's only BT cotton that has been allowed to grow in the fields, and no other transgenic crop has yet been approved for growing in the fields in India. But the only crop, but it means BT cotton. I think India is the first or the second largest producer of BT cotton in the world. Means it's in very large fields that BT cotton has been allowed to be used, to be cultivated. Now, coming to the safety concerns, one concern was that apart from your gene, so whenever you have to make a transgenic plant, then apart from your gene of interest, because of the selection process, like for making any this transgenic organism, you need to have a selectable marker. So one concern was that whether this selectable marker that is going in the crop, will it affect human health or not? So could it be that, let's say, kanamycin gene, it goes inside the human body? Does kanamycin resistance gene by itself can cause any, means whether humans will be allergic to it and all. So one is like what effect it will have on human health. And apart from that, another concern was that even if it is not of direct impact to the human health, but there is a chance that this gene is transferred to bacteria in the human gut. And then finally, is there a chance that this gene can get, spread to the environment and all? So then the concern was, there was one way that was found by which we can remove the selectable markers after you have made the transgenic plants. So that can be done by cloning, by ensuring that your antibiotic resistance gene is flanked by these Cree sequences, or let's say lock sequences, or let's say Cree recognizing sequences. So these sequences, there are around 34 bases, And once your transgenic plant is made, once you have selected that, okay, this plant has been made, then you can get rid of this gene by activating the Cre recombinase. So in the transgenic plant, you also add the this Cre recombinase gene, and this gets activated, or let's say this gets expressed, and then it recognizes these sequences and removes the intervening sequence. So as a result, what we will finally have a transgenic plant which will have your gene of interest, but the selection marker has been removed. So this is one strategy that was used by which you can introduce, you can make a transgenic plant which does not have any gene of concern, and that is removed by Cre recombinase. And another thing is that to ensure that your final plant also does not have this Cre recombinase, you look for the segregation pattern. So you uh, you grow these plants and then you cross them with themselves. So in the progeny, there might be a case that you get a progeny plant in which you have your gene of interest but the Cre recombinase is lost. So similar strategy can also be used in which, let's say, you don't use the Cre recombinase and you have your gene of interest and your kanamycin gene as two separate let's say recombination events in two different vectors. So you have to select for a plant which has your gene trait as well, I mean whatever gene fragment that you have added, and you have let's say kanamycin which is introduced in another chromosome. Now when you cross this plant among itself, so there can be a this segregation pattern in which you can get a plant in which only your gene of interest is there while the kanamycin gene is lost. So finally, by some method you have to find, maybe by southern hybridization and all, you have to look for plants which have only your gene of interest but kanamycin gene has been lost because of segregation patterns and so on. And another point with respect to the this safety concern, I mean, sorry, with respect to the Cre recombinase, where it is used by the developers of these transgenic plants, by the various companies which were making it, is one technique called as terminator technology or genetic use restriction technology. So this technology, this was developed by the various companies which were making these transgenic plants, by saying that, you know, by this technology they are ensuring that the genes will not be spread in the environment, but actually it came with one important point, that this they ensured that the plants, the transgenic plants that they are growing and the seeds that they are selling to the to the farmers, so farmers will be able to grow these plants but they will not be able to harvest the seeds, I mean their seeds will be sterile in nature. So it meant that every year the farmers have to buy the seeds from the developers, from the companies. So one way it was like not liked by the farmers at all, because then they became totally dependent on the companies to buy the seeds every year, so which was not a good thing. So it is a highly controversial technology, and I'll tell you that how smartly these company people had developed this technology. So here, what they do is that the transgenic plant that they develop, they have that plant definitely has that gene that they transfer. Let's say any, you know, herbicide, glyphosate resistant gene or insect bt toxin, whatever gene that they have cloned, so that gene will be there. But apart from that, there will be two other genes. One will be the rip gene. What is rip gene? It is a ribosome inactivating protein, means a protein which will act on one of the rRNA and you know, cleave it. So as a result, yes. Uh,
Ma'am,
yeah.
ma'am, in the previous case, um, so when we were like when Cree was also introduced into the plasmid, in that case, would it already like since the antibiotic was resistant gene was already flanked by logs, would it already remove the antibiotic resistance before we can select the uh, I mean the
No, no,
plasmids that
yeah,
are
yeah, yeah. A very important point that maybe it, completely removes it before only. But for that,
Mm,
you use this inducible promoter, like in
agree.
the
Okay.
current case that I'll tell you.
Okay. Okay.
Hmm. So use inducible promoter so that you can control the expression of free recombinase. Any other question for this particular thing? Okay. So in this case, what they had done was, yeah, so your gene of interest is there, and this RIP gene is there, which acts on ribosome and then cleaves it. And as a result, the protein synthesis is completely blocked. And as a result, the cell which has this high expression of RIP gene, it will die. Okay. So the plant that they develop will have their gene and a rib gene. The only difference would be that the rib gene will be blocked by some blocking DNA. Any stretch of DNA is put in between, which is flanked by the Cre site. So this pink thing is whole rib gene. Only thing is that it is blocked by a particular gene sequence, which is flanked by these Cre recognizing sites. So it means that RIP means the transcript will be made, but because it is blocked, the protein, the active RIP will not be made unless it is acted upon by Cre recombinase. So another gene is there which is cloned, which is Cre recombinase gene, and it is under control of tetracycline promoter. So when let's say you treat it with tetracycline, so this promoter will become activated and Cre will be made. This blocking DNA will be removed because this will recognize Cre sites, remove this, and what we get is a functional RIP gene. Okay. Now, the thing is that the developers, I told you they will have their gene of interest, this gene and this gene, but their plant will grow normally and will have normal seeds produced because after all, their RIP is inactive. So for them, there is no problem. They will grow their gene and they will make the transgenics. They will harvest the seeds and finally they can keep growing and selling it to the farmers. Now the only difference was that before selling it to the farmers, what they did was that they treated the seeds with the tetracycline. So what will happen that the seeds that will develop, they will have this the seeds will have this uh, intervening region removed, and we have a functional rib gene. So it means that the seeds which are being sold to the farmers will have seeds which have functional rib gene, and as a result, will they be able to grow the plant? Yes, because this rib gene is under control of a promoter, this embryo specific promoter. It means that in any other plant, when the plant is developing and all, the gene, the gene will not be transcribed because it is only embryo specific. So once the whole plant is made in the field, means the farmer grows this seed. I mean, the farmer grows this seed, the plant will grow normally. It is only when the seed is developing in their plant that this gene become active because it is embryo specific promoter, and then the rib gene will be made. As a result, the seeds that will be made, they will be highly sterile in nature. So it is said that even though some seed is made, and the if the farmer grows that in the field, so it will be sterile and they will not be able to grow the plant. Is this clear? It's a little complicated, but means I just want to say that the developers, they are not having problem in growing the plants and harvesting the seeds every crop season, because after all their rip is inactive every time, right? It is just that some a bit of seeds that they harvest, they incubate it with the tetracycline. As a result, the cree will get activated and remove the intervening region. So the seeds that the farmer gets has this full, or let's say unblocked rib gene. So in a way, if it gets expressed, it will give a active protein. But the plant can easily grow because the promoter is highly specific in nature. So during any stage in the plant, it will not get activated and interfere with the growth process. But when the plant is having the seeds, then seeds will be highly compromised, because the rib gene will get activated, because we have put a promoter which is highly specific for embryo, and as a result the seeds will be sterile. So it is because of this reason that this terminated technology, it was resisted by various farmers. Is it clear? Okay.
Yes, ma'am.
Mm. Okay, so I'll come back to another issue with respect, with respect to the safety concern. I just want to tell you that which are the various regulating, because these are transgenic crops. So three different agencies have to be very prevalent while approving their use. One would, of course, be agriculture, the Department of Agriculture. One would be the environment, because there is a chance that the transgene that you are putting, there are ethics related to it. So if it gets spread in the environment, will it affect the wild type crops and so on? So environment is definitely another agency from which you have to take the approval. And the third is food and drug administration, because of course it is a crop and many of these are used as direct food for the humans. I mean, not all, like cotton is not directly consumed by the humans, but many fruits or crops, they are consumed by humans. So obviously, Food and Drug Administration agency also is required to give the approval. So the approval of any transgenic plants takes, several years, and also development of these genetically modified plants and the approval, it requires a lot of money. So yes, these are the, I wanted to sensitize you to the fact that there are various government regulations regarding their use. And in India, GAC means IBSC. One is the Institute Biosafety Committee, as well as there is the full name is, I think, Review of Genetic Modulation and Genetic Engineering Appraisal Committee. These are the committees which are required from these people. You have to take the approval for growing the transgenic crops. Now, this example I've brought directly from the T.A. Brown book. It is a good example. It was done after a lot of studies of these various transgenic crops that had been developed. So this study was done in Europe, I mean England, Wales, and Scotland, for these transgenic crops, in which these crops, they were grown in a field as opposed to another field where non-GM crops were grown. And then, so means there was one field where there was, let's say, this glyphosate resistant crop, or whatever GM crop you had used, and another field was there where non-GM crop was grown, means the original crop. So then they compared the two crops after growing crops, these fields actually, they then monitored these fields. And what they found was, this is, I think that you please go through it, that so in nutshell, what they found, that the animals or the butterflies, these insects like butterflies, bees, etc., their their population varied in the two different fields. Why? Because many of these, they thrive on the seeds of the weeds. So one thing is that it is not directly related to the the gene that you have added, because of which this insect population changes, but it is because these GM crops, for example, in this case herbicide resistant crop, because of that the weeds population was different between the two different field. And the seeds of the weeds were probably eaten or affect, they were they were used by these various insects, because of which they found that the insect population varied between the two fields. So that's what they have written. They arise because these GM crops give farmer, and also the farmers get new options for the weed control. They may be using different herbicides, or they may be applying the herbicides in a different way, because of which, the diversity between the two fields, it was found to be different. So what ultimately I want to convey is that it does have some ecological impact. And also there are chances that the gene from the wild type variety, sorry, from the GM crop, it may be transferred to the non-GM crops, because there is a chance that there is cross pollination or something, that the gene which we are using, let's say for making a better, or let's say highly resistant variety, it gets passed on to the wild type or weeds also. So then what we'll have is highly resistant and very robust weeds, which we do not want. Or let's say during course of time we may lose our wild type crops, and they may become contaminated with these transgenes that we are putting in the environment, and so on. So there are a lot of issues related to GM crops, a lot of politics also, and a lot of discussion, a lot of beliefs also, and a lot of misinformation also, because of which in many countries they are completely banned still. They are not used at all, and in many countries they are very prevalent, which have done a lot of risk assessments, and they are quite convinced that use of GM crops for these purposes, or only these GM crops, can definitely be used by the humans, and while others, they still require approval, and so on. So if you want to have some idea which countries are using which GM crops and so on, you can go to some recent papers, or maybe even Wikipedia, which has updated which countries are using GM crops and all. So if I come to the, the concerns with respect to GM crops, one could be the transfer of these transgenes, or even antibiotic resistant genes to the environment, change in the biodiversity, contamination of the wild type varieties, which are native varieties, by these transgenes, and getting some new crops and so on. And why we may lose those wildlife varieties, because maybe they are slow growing and whatever, and or they are less resistant and all, so they might be taken over by these transgenic crops. And one is that I told you that in case this terminator technology is used, one option is use of this method, and there are various different methods of terminator technology which is used by various different companies. It's not limited to only this exact method of altering, but many other methods have been used by various companies to ensure that these farmers are totally dependent on them. So because of this, there is a economic impact as well. So, and social impact as well. So why these impacts are there? Because obviously for farmers it will be very, very difficult to buy the seeds, which are very expensive as compared to regular seeds. And social impact because let's say even in the fields where they had introduced these herbicide resistant um, what was that or let's say insect resistant and so weed resistant, this herbicide resistant crops. So there were laborers which were employed to remove these weeds. So their job was gone. So means lot of studies have been done to even assess the economic impact of growing these transgenic plants. So if you see in the development of that transgenic maize study also, I think I've given a link to that. You can see that there were various studies which were done, and even economic impact and social impact was also considered a very important concern for these GM crops. So that's all for today, and in fact for this whole course. So I'm open for questions. Okay. So there are no questions?